En nombre de la Vicerrectoría de Investigación y Posgrado y del Centro de Investigación de Estudios Avanzados del Maule, les damos la más cordial bienvenida. Esta charla magistral se enmarca en la inauguración del programa de Doctorado en Salud Ecosistémica de nuestro Centro de Investigación. Saludamos a las autoridades presentes, doctor Claudio Rojas Miño, rector de nuestra Casa de Estudios, doctora Liliana Zúñiga Venegas, directora de Investigación, doctora Karina Vilches Ponce, directora de Posgrado, doctor Hugo Benítez de la Fuente, director del Centro de Investigación de Estudios Avanzados del Maule, académicos, investigadores, estudiantes, colegas de nuestra Casa de Estudios. Saludamos también a nuestro expositor, doctor Leo Buquebum. Y para dar inicio a esta actividad, invitamos a nuestro rector, doctor Claudio Rojas Miño, a brindarnos unas palabras.
Bueno, buenos días, creo. Sí, buenos días a todos y todas. Saludos a nuestro invitado, el profesor Leo Buquebum. Ya le di mi excusa de que no lo voy a poder acompañar todo el rato, pero sí voy a, por lo menos los primeros 20 minutos, media hora, lamentablemente tengo otra reunión. Pero me siento muy congratulado, contento de que en el marco del programa Doctorado recién salido al horno, por decirlo de alguna forma, de Salud Ecosistémica, un programa muy novedoso, muy potente, que probablemente vamos a tener que cuidar y cautelar mucho en términos de cómo se desarrolla, pero evidentemente es un programa que por sus contenidos y por lo que promete es realmente un programa de formación de muy buen nivel, por llamarlo así, y muy en la frontera del conocimiento, y no solamente en la frontera del conocimiento, sino que en la frontera de los problemas que hoy día estamos teniendo como sociedad global, en realidad. Así que, dicho eso, que les vaya muy bien, y muchas gracias a nuestro invitado. Solo eso.
Muy buenos días, muy bienvenidos a todos, agradeciéndole la presencia acá. Estamos muy contentos de inaugurar el año académico de nuestro 10º programa de doctorado. Vino a cerrar el ciclo, el proceso de ejecución del plan de desarrollo estratégico desde posgrado, entonces es un hito muy importante lo que se ha logrado. Acabamos de cerrar también la entrega de becas doctorales, estamos en un proceso ya de instalación de la formación doctoral. Les comento también que próximamente vamos a tener nuestra primera ceremonia de graduación y entrega de medalla doctoral el 27 de abril, así que también ahí vamos a invitarlos a todos, estamos en proceso de organización, agradecerle al invitado internacional y comentarles que el programa de doctorado de salud ecosistémica empezó a diseñarse más o menos al inicio del Centro de Investigación de Estudios Avanzados del Maule el año 2020, de haberse iniciado el diseño el mismo año. Costó un poquito sacarlo adelante por el concepto que lleva detrás de esto de la mirada integral de la ecología de los animales, del medio ambiente y de los humanos, y el concepto del One Health que hay detrás de su formación. A nivel comparativo, a nivel de análisis internacional, uno puede ver que este tipo de programas ya está muy instalado, muy presente en los sistemas de formación de capital humano avanzado, tanto en Europa como en Estados Unidos, pero que en Latinoamérica todavía está predominando un poco un concepto más antiguo respecto a los programas doctorales, que tiene que ver con el campo disciplinar, con el desarrollo más bien de un ámbito disciplinar y no basado en la resolución de problemas usando diferentes metodologías y herramientas dependiendo de la disciplina con la que abordemos el problema de contexto que nos manda. Es un poco nuestro sello, nuestros 10 programas de doctorado tienen este sello interdisciplinar y que además interfacultad, entonces salen un poco del nicho de lo disciplinar y están enfocados en generar investigadores que van al servicio de la comunidad y que están resolviendo potentes problemas que son de interés público y que en el futuro pretendemos que impacten también en el desarrollo del medio productivo, por ejemplo, a través de nuestros nuevos programas de doctorado que son más bien de orden tecnológico y que van apuntando también a equilibrar el desarrollo de cada área emergente de nuestra universidad, ¿ya? Tiene toda una estructura. Vamos a partir, yo creo que con 4 o 5 estudiantes ya, por lo tanto estoy muy contenta. Tuvo una gran convocatoria a pesar de los plazos con los que se dieron. Hubieron muchos postulantes, así que es un programa que va a ser muy exitoso, que amplía además las posibilidades de desarrollo de los académicos y académicas a través de la guía de tesis y finalmente de poder consolidar su carrera como investigadores dentro de nuestra institución. Agradecer al director del programa, al director del CIEAM, al comité académico que trabajó para lograr que en este momento estemos inaugurando este programa, a los administrativos también, profesionales del CIEAM que apoyaron, también a los de la dirección de posgrado que apoyaron en su diseño, y muchas gracias por estar presentes. Espero que sea una jornada muy llena de sorpresa, de conocimiento, y que sea muy productiva para todos. Muchas gracias.
Agradecemos las palabras de la doctora Karina Vilches. Dejo con ustedes al doctor Hugo Benítez de la Fuente, director del Centro de Investigación de Estudios Avanzados del Maule, CIEAM, quien compartirá unas palabras y dará la bienvenida a nuestro invitado.
Gracias, Miguel. Buenos días a todas y a todos, señor rector, directora de posgrado, directora de investigación. Para mí es un placer estar ante ustedes en un día muy importante para nosotros. Para nosotros es la inauguración del Doctorado en Salud Ecosistémica, un proceso que fue intenso, largo, desde la creación del CIEAM. Se generó la propuesta de poder elaborar un programa de doctorado, y este programa de doctorado tiene un matiz súper interesante, que, bueno, primero es el primer doctorado en el tema en Chile, ya hay varios doctorados esto a forma internacional, pero que además trae la integración de líneas de investigación desde una visión One Health, que es una salud que trae salud de animal, salud ambiental y salud humana. Además, este doctorado trae también la integración multifacultades. Es un doctorado que, si bien nace del Centro de Investigación de Estudios Avanzados del Maule, del CIEAM, tiene un apoyo importante de todas las facultades, diferentes facultades de esta universidad, lo que nos demuestra que en el proceso a la integración y a la acreditación institucional en investigación y en posgrado que viene, nuestro paso es poder trabajar en conjunto y poder generar ciencia de calidad en un programa donde estamos a nivel de punta como país, principalmente que ahora en tiempos de cambio climático, poder entender y comprender las relaciones del ambiente y cómo su distribución con los animales está, viene súper interesante. No me voy a alargar mucho más con esto porque quiero presentar a nuestro conferencista magistral, el doctor Leo Bekkeboom. Él es de Países Bajos, Holanda. Él es investigador, es entomólogo, investigador en biología evolutiva. Tiene aproximadamente cerca de 200 artículos científicos publicados, autor del libro The Evolution of Sex Determination. Él trabaja principalmente en aspectos de la selección y reproducción en insectos. Tiene un amplio currículum en entomología, ha trabajado en diferentes grupos de insectos, partes de control biológico y buscando técnicas para poder comprender la integración del estudio de la reproducción a nivel general. Hoy nos va a entregar una charla. Él la preparó principalmente para la inauguración de este programa de doctorado. Tuvimos la suerte de que accedió a nuestra invitación. Él anda con una agenda muy apretada. Participará en Talca solo el día de hoy y ya se vuelve a Santiago en parte de su agenda. Así que ahora voy a usar un switch a inglés para poder presentarlo. Él no habla español, pero por supuesto, después de la charla, cualquier pregunta que esté asociada a la presentación la pueden hacer en español y yo puedo ayudar en la traducción. So now you cannot freak out very much Spanish now. I will change to English. So for our university, we are mostly very happy to having you here with us. It's a pleasure for me to present your talk. Leo will talk about the insect reproduction, fundamental research and applied aspect. Leo University is now having the inauguration from our PhD program in ecosystem health, and they are all happy we're here. The Center of Studies, the Research Studies, Advanced Studies in the Maule, is the centers that allow the program, but it's a program that also have a mix of different faculties in this university to try to go to one health idea to understand the health of animal, human, and the environment. So for us, it's a pleasure to having you, and please come here and give us your plenary talk. Thank you very much. Gracias a todos.
Well, thank you very much. Yeah, I'm very excited to be here. First time in Chile, first time in South America for me. So that's already very exciting. I'm honored to be able or to be here to be at this opening of this new program, and I'm also very thankful for also for you, Hugo, for inviting me. So what I'll do is I'll give it a talk, a bit general talk about some of the research I've been doing over the many years that have been an entomologist. And there will be both fundamental and applied aspects to this. I started as a fundamental scientist, as an evolutionary biologist, interested in fundamental questions, trying to explain why we see certain biological phenomena the way they are at the current time. But then also over time I became more and more interested in applied aspects of the research that I'm doing. And this was not just interest, it was also a little bit sort of forced, because there is just more funding, financial funding for applied research, especially also when you work with insects, than for fundamental research. But I basically do both. Okay, so I have a... no, okay. Oh, yeah. I've just one slide about myself to maybe a little bit further introduce myself. So I'm an evolutionary geneticist and an entomologist, and I work in this building, which is the Groningen Institute for Evolutionary Life Sciences of the University of Groningen, and Groningen is right there here. For those of you who may not know, this is the Netherlands here, a very small country here in Europe, and Groningen is in the north. It's only 200 kilometers north of Amsterdam, which you probably all know. It should be this one. No. Okay, let me check. Okay, so my major research lines are on the genetics and evolution of reproductive systems, sex determination mechanisms, and life history traits in insects. And as I said, both from a fundamental and applied perspective. And you see here some of the organisms I work with, and some topics of research that I'm doing. I'm not going to mention them all. And I now also collaborate with some companies that you see here at the bottom. I'm also an editor-in-chief of this journal, which is called Entomologia Experimentalis et Applicata. So that's still an old Latin name, as you may realize. And the journal is about 65 years old, and it's basically the scientific journal of the Netherlands Entomological Society. And no, should I just hit? Yeah. Okay. Yeah. I can also stand there, I guess. It's easier, right? Okay. Maybe I should stand there, right? Yeah. Oh. If I stand here, is it easier? Okay. Oh, now it is doing something. Yeah, okay. Okay, so just first a few very basic information about reproduction, insect reproduction. As you probably all know, There are several forms of reproduction. Of course, there's sexual reproduction, where we have males and females, and they produce haploid gametes, and they fuse, and they produce a new individual, like what happens in us humans, of course. But there's also what is called parthenogenesis, where there's females that only produce daughters, and there are basically no males. And this can be done in two ways, via haploid or via diploid gametes. This is called automixis, and this is apomixis. This is more for the animal world, and that's more for the plant world. So parthenogenesis is basically the development of an individual from an egg without any fertilization. And then there's a third form, which we call fission or vegetative growth in plants, where basically you can just sort of cut off a part of your body, and that part grows into a new individual. And of course, this is impossible in humans, as you will all know. But many plants do this, but also many other organisms. So here I show just a strawberry, which can just multiply by vegetative growth. But there's also other invertebrates here. You see hydra and flatworms. If you cut a piece off, then that piece of body just grows into a new individual. Now, not in insects, though. Okay, so this does not occur in insects. So for the rest of the talk, you can forget about that. But these two forms we do find in insects. Then, of course, another sort of very basic distinction between individuals is whether the male and the female reproductive organs are in the same individual. And then we talk about a hermaphrodite. And in fact, 96% of all plants are hermaphrodites. But of course, we have also organisms with separate sexes, again, like us humans. This is called dioece in plants. So we basically have separate male and female individuals. And here you see the groups in the animal kingdom that have the separate sexes, which is called gonocorists. And these are also the groups that we're maybe most familiar with, like the mammals, the birds, the fish, and the insects. But there's many, many other animal groups where almost all individuals or species are hermaphrodites. Okay? You should realize that. But again, not in insects. So in insects, there's no hermaphroditism, and we have always two sexes. Now, if you then look at a little bit more variation, a bit more sort of down to the genetics of that variation of the two sexes, it turns out that the process of sex determination, so how the two sexes are genetically determined, is enormously diverse. And this may come as a surprise because the end product is always the same, right? Males and females. But apparently, the way how these males and females are genetically determined is very diverse. And we really don't understand we still don't understand very well why this is so diverse. Because other sort of basic developmental processes, like embryological processes, they are rather conserved over evolutionary time, meaning that the same genes are involved in say, the lower organisms and the higher organisms, if I may use that term. But sex determination, for some reason, is extremely diverse and also changes quickly in evolutionary time, apparently. So this is sort of illustrated here with the insect orders, and the different colors are basically different sex determination systems. We have in dark blue, as you can see there, what is called male heterogamety, which we as humans are also, where females have two equal sex chromosomes, two equal sex chromosomes, which are called the X chromosome. Of course, females are XX, and males are XY. They have the two different sex chromosomes. This occurs in all mammals, but also in many insects, as you can see there on the right. But there is a variant of that. There's also what is called the XO system, which is basically almost the same, but there is no Y chromosome. So the O basically stands for null, for nothing. Okay? So males and females only differ in that females have the two sex chromosomes, 2 copies, and the male have only 1 copy. And you also find that in many insects. And that's the light blue on the right side. And then we have the orange groups here. So these are two separate groups. Like, for instance, all the butterflies, they have what is called female heterogamety, where it's the female that has the two different sex chromosomes. And we then indicate that with stat W, whereas the males have the two equal sex chromosomes, and they are ZZ. And then finally, there are yet other, you see another color here, the green ones. So these are, for instance, the Hymenoptera, to which I come later. These are all the ants, the bees, and the wasps, more than 1 million species worldwide. And they all reproduce by haplodiploidy. So they do not have sex chromosomes. The only difference between males and females is that females have all chromosomes twice. They're diploid. And all males have all chromosomes once. They're haploid. So there's no... Males and females carry exactly the same number of genes, exactly the same genes, but only females carry them twice and males only once. And that, of course, already poses an interesting question, is because if you have if all your genes only once, how can it be that you become a male, whereas if you have all the same genes twice, you become a female? And we will look at that later on. So this is just to illustrate you some of the variation. I think I have one more slide to even show you a bit more variation. And this is beautiful work by Ficoso and Bachtrock that have been sequencing lots of genomes. And then they found out that the sex chromosome in one species is not the same sex chromosome in another species. And so this is what is called the karyotype of Drosophila, and with 4 pairs of chromosomes. And these chromosome arms are all indicated with a letter. So these are called so-called Muller elements. And you see here, here I have colored the different elements. And now here what you can see is all different species just in the flies, the order of Diptera. And all the different colors indicate that the different chromosome is the sex chromosome. So each time a different color, you see there is the male determining function is on a different chromosome. So even within this male heterogrammatic system, you see a lot of variation in which chromosomes are actually sex determining. Okay, so that's a bit of a sort of general background. So now we'll start to ask some questions. And one question that sort of came up during my research is, can we change population sex ratios? So can we somehow manipulate the proportion of males and females in the population? As we will see later on, these ratios are usually equal. Like in humans, again, there's about equal number of males and females. If we, maybe not in this room, but if we look at the whole world population, then it's really almost 50-50. But for insects, that's the same for insects, but for insects it can be interesting to try to manipulate the proportion of males and females. And that's because for certain applications in entomology, females or males are of more value than the other sex. And one example here is for feed and food production. They should say food, excuse me. So feed is basically food for animals, and food is used for food for humans. That's the difference between feed and food. And this is an enormous upcoming industry. There's a lot of research currently going on, a lot of financial support to look into this, to see whether we can culture insects to basically to produce food, because they come with a lot of benefits. They have very short generation time. You can grow them on all sorts of waste streams, and they don't put such a lot of pressure on the environment, like, for instance, the traditional livestock, like cows and chicken. So, and then it becomes interesting if we can produce more females, because only females lay eggs, and you can have one male mating with multiple females. So if you have more females in your production population, you can reach a higher production. So this would be economically benefit. There are other important entomological techniques where it would be useful to change the proportion males and females. For instance, in this very important technique, which is called the sterile insect technique, is where people mass produce insects, and they sterilize then the males of these insects, and then they release the males. And these released males, they compete with natural males in the population, and in that way they can reduce pest populations. So for food production it would be useful to have more females, but for this pest control technique it would be useful to have more males. And we are involved in one program of the FAO to see whether we can do this. I'll come back to that later. And then a final example is biological control. You're probably familiar with that where you use certain insects that eat other insects, and you apply them, say, in greenhouses and in crops to kill other insects. And in that case, again, especially when we use parasitic wasps, it's only the females that parasitize other insects. They lay their eggs in other insects. The males don't do that. They don't lay eggs, so they don't kill other insects. So in that case, it would be very useful to have more females. Now, but that's not so easy to change sex ratios of populations. And in fact, we know that already for almost more than a century. And that is because there is what is called sex ratio selection, which is called Fisher's principle. And that is a evolutionary process, that sort of natural selection process that leads to always equal males and females. And I'll explain you in a moment how that works. So the term sex ratio here is usually the ratio of males and females. It's usually given as the proportion males. And as I said, it's usually equal in populations. And of course, that is partly because of the sex determination system, which I have just introduced to you. Because in this male heterogametic system, if you have females and males XY, of course, as you all know, if you're a male, you produce 50% of your gametes which has the X chromosome, and exactly 50% that produces the Y chromosome. The Y chromosome is male determining, and if you don't have a Y, then you become a female. So this is basically a genetic way that is sort of responsible for this equal 50-50 proportion. But that's not everything. And that is something that Sir Ronald Fisher already realized in his book of 1930, so almost a century ago. There is also what is called natural selection that acts to keep the frequencies of males and females equal. And that is shown here, a very simple sort of graph here. So here's the sex ratio, the proportion males. And the idea is that if there's more males, then the fitness of a son is lower than the fitness of a daughter. Okay, so any sort of mother or pair of parents, in a case here where there are more males in the population, if they produce more daughters, they have a fitness benefit. Okay, so those individuals will therefore then increase in the population, and therefore the number of daughters will increase, and then till again the number of sons and the number of daughters, or males and females in the population, is equal again. And this is what is called an evolutionary stable strategy, which is basically a phenotype in a population that cannot be replaced by a mutant one. Okay? So basically natural selection always acts to go to this point here. And again, equal numbers. So I mentioned these sex chromosomes, therefore they prevent easy manipulation of population sex ratios. And what I show here is a table that we put together for a special issue of this journal here that I introduced to you before. And here what you see is sort of the main insects that are being used for food and feed production. Of course, we started with the honeybee, which you all know produces a lot of honey for us. But then here you see a whole bunch of crickets, so orthopterans, grasshoppers, but also beetles that are being used for food production. And for feed production, you see here a number of flies, to which I come back later, and again some beetles. And if you now look at the sex determination system of these groups, then you see that indeed they all have these sex chromosomes, except for the honeybee. I'll come back to that later. So some of them have these XO systems, like all the orthopterans, all the grasshoppers and crickets. They have an XXXO system. Beetles have both of these systems. Flies have typically this system, and these cockroaches also have this XXXO system. So, so we are dealing with the fact that having such a system that sort of constrains our possibilities to manipulate the sex ratios in populations. So we are part of a consortium now where we are trying to look more into this, which is called Insect Feed. This is funded by our National Science Foundation, and this is together with colleagues from Wageningen University, which is in fact the agricultural university in our country. And our colleagues in Wageningen, they focus on this species, which is called the black soldier fly, which is basically used worldwide now already to produce animal feed and also proteins. Okay. So there are quite a few factories now worldwide already where these flies are produced en masse. But we focus on this fly, the common house fly. You're probably all familiar with it. I think it occurs worldwide, called Musca domestica. And we try to, rather than killing this fly, if we see one, we basically culture them. We produce a lot of them. And the idea is to feed the larvae of these flies to the chicken. And the chickens really like these live larvae as food, I can tell you. So then, so we basically then use this housefly as a sort of a model system to see whether we can manipulate these sex ratios. Now, how could you do that? Of course, you could say, okay, let's just split males and females, and we only put the females into the mass production. But if you're talking about millions or millions of flies, then of course it's an awful lot of work to just separate all the males and females. Yeah. So that's a problem. So if you would just select them on phenotype, it's of course you can make mistakes. You're dependent on that you can easily distinguish males and females in the species, which is not always the case. It's of course enormous time consuming and labor intense. So what we looked at to see whether there were certain phenotypes, so in which males and females differ, that we could use. So here just a very simple sort of example where we just look at adult body size. Okay. And yes, females and males are slightly different in that. So the females are here, this is at 25 degrees, and this is at 18 degrees. Females are slightly bigger than males. But of course, as you can already see here from the variance, there's a lot of overlap. So you cannot just use body size to separate males and females. Yeah. So that's a problem. Also, what you would like to do this as early as possible. So not in the adult stage, you would like to do this in say in the egg stage, ideally, because if you can already make the selection there, you don't have to grow the other sex in which you're not interested in. So that will save a lot of time and money. In some flies, there are males and females there are two types of puparium colors, but the problem here is, and then if that would be the case, this is not the case in houseflies, but I guess you could make a mutant, but the problem is then this must always be the males and these must always be the females. So if you have such a color polymorphism, it still doesn't help you if it's not sex linked. And of course, if you look at the larvae, we really can't see whether a larva is a male or a female. I mean, they all look identical. But then we found out that there's an interesting phenomenon in flies, and this has also been found in mosquitoes, for instance, which is called protandry. And that basically means that males have a faster development than females. Okay? So what you see here is development of these houseflies at 25 degrees here, and here it's at 18 degrees. I just look at the top figure because the results are the same. And here you see that at 25 degrees, the generation time of these flies is about 11 days. And on day 11, the first flies come out, and they have a nice rhythmic emergence. So they only emerge during the light and not during the night. But on day 1, you see that there's more males coming out than females. And that's still a bit on the second day of emergence, day 12. But the later emerging flies are more often females. And then we thought, okay, if we now just take the first emergence flies out of the population, right? We can just do that. And then we could show that if you just focus on the later emerging flies, you get about 55% females. So that's already a 5% increase just from phenotypic selection. And we're in fact now sort of seeing whether this works in a mass production scale. So we're collaborating with this company that builds these sea containers, which contain about 6 million houseflies, and they produce about 3,500 kilos of larvae per day, and these are fed to the chicken. And we're seeing whether we indeed can increase the production by taking the early emerging flies out of the population. Now then, what we realized, we then thought, being geneticists, can we artificially select for this protandry? So can we select the flies in such a way that males develop even faster and females even slower, because then we can sort of separate males and females more and more based on their developmental time. So this master student in our lab, he did 6 generations of selection, where he took males that emerged early and he mated them with females that emerged late. So males from day 11 with females from day 14. If there would be a genetic difference between them, we may be able to select for that. And this is in fact what people managed to do with mosquitoes. Okay. Exactly the same procedure. But in flies, or in houseflies, it doesn't work. So what they show you here is what we call the protandry index, which is basically the developmental time of the females minus that of the males. And the females have a longer developmental time, right? So the index is positive, as you can see here. And the black dots are the controls that were not selected. Okay, so we just had random mating of females and males, not depending on which day they were emerging. And the selection ones are always these early and these late ones. And as you can see from the distribution of the orange and the black dots, there is no response to selection at all after 6 generations. So unfortunately, this did not work. Okay, then we also use these houseflies from another perspective. And now I come back to this sterile insect technique, where I said earlier, if you're interested in mass producing flies for this technique, you want more males, because you want to release the males and you don't want all the females. Right? Of course, you need some females to keep the populations going, but you don't want to sterilize all these females and release the females. You only like to get males. Okay? And this is the sterile insect technique. I'm not sure whether you're familiar with this, but yeah, basically I think I've already more or less said it. So you mass culture a certain pest insect, and then you irradiate the males, or you irradiate them all, and then you release the males, and then the sterile males. So you release so many males, then more than that are out in the natural populations, and then the idea is that the sterile males compete with the natural males to mate with the females, and of course when a female mates with a sterile male, she will not produce any offspring. Okay. And this is a very successful technique, and in this way people have really irradiated certain insects from certain areas, like the screwworm, I think from Mexico down to Panama, has been completely irradiated with this system. And this is, I think, the largest probably insect plant in the world, which is in Guatemala, where they produce millions and millions of these Mediterranean fruit flies that are a pest of citrus fruits. And they release these every day, and in that way they protect their crops. So from there is a lot of interest and also financing, again, from the FAO, the World Food Organization, to further improve these techniques. And there's a special program in there where there's funding for trying to find out how we can manipulate the sex determination system of these pest species to produce more of one sex. And this is some work I do together with Daniel Bob in Zurich. And again, we use these houseflies to see whether we can do this. To explain you how that works, I have to tell you a little bit more about the molecular basis of sex determination in insects. Females, again, are XX, males are XY, and there's typically a dominant male determiner on the Y chromosome. So a gene, if you have it, you become a male, like in humans. It's the SRY gene on the Y chromosome. And if you don't have it, then you become a female. There is, in insects, there is a certain gene which is called transformer, which is already transcribed during oogenesis. And the messenger RNAs of that gene are put into the egg by the mother. And that basically needs to be present to start the transcription of the transformer gene itself in the zygote. So when this maternal contribution is not there, then this loop of self transcription cannot start. But if it can start, then this gene is what is called sex-specifically spliced into a female mode, and that then turns on this gene, which is called double sex, and it's also spliced into a female mode, and this is a transcription factor that then starts all sorts of other genes downstream that are responsible for female differentiation. Now, of course, if the egg is fertilized by a sperm with a Y chromosome in it, and the male determining factor comes in, then what happens is this male determining factor sort of breaks this loop, this autoregulatory loop, and therefore transformer is not spliced into the female mode, but into the male mode, and therefore double sex is spliced into the male mode, and therefore male differentiation follows. So what we set out is to try to identify the male determining gene from the housefly. And when we started this work, like 5, 6, 7 years ago, there was not a single male determining gene known from insects. Okay, so nobody knew how males were made in insects. It was clear that it's not our male determining gene, because that's not present in insects. That's only in mammals. So this took quite a bit of work. This is our PhD study of Akash here. And at some point we managed to find the gene and characterize the gene. And I'm showing you here the gene, and we call it Musca domestica male determiner, or MDMD. And it turns out that it's very similar to another gene in flies, which is called mucampholin, and the name comes from Drosophila. But it's also called CWC22 because it's also present in mammals. Okay, so we also carry this gene, but this MDMD gene is slightly different from this mucampholin gene, and this is a very important gene, a central gene in the so-called spliciosome. So it plays an important role in enzymes that are responsible for splicing of genes. So we were quite surprised that it's some sort of a duplicate of an existing gene. And then we start to look whether we could find this gene somewhere else. And it turns out, no, we can't find this gene anywhere else. We looked at the most close relatives of houseflies, and it wasn't there. So this seems to be a gene that is specific for houseflies. Here I just show you that indeed this NCM gene, it's present in both males and females. So here we have males and females from different strains, and but the MDMD is always only in males and never in females. Okay. So this gene, the original gene, is expressed in both sexes, but this copy, which now has become a male determiner, is only present and expressed in males. In the meantime, so we published this in 2017. and we thought we would be the first one ever describing a male-determining gene from an insect, but unfortunately we were beaten by some other groups, and of course these are groups that work with mosquitoes, because there is more money to work on mosquitoes than on flies, unfortunately. So there were 2 publications just in the years before on mosquitoes. And they found here the Nix gene, and here they call it the Job gene, and we have MDMD. And then after us, there was also the gene found in the Mediterranean fruit fly. And then, of course, we compared these genes, and it turns out there is just no homology between them at all. These are 4 completely different genes. Okay, so we find a gene in one insect, but it doesn't mean anything about whether we can find it in another insect. And if you know that there's more than, I think, almost 3 or 4 million insects, and if each insect has its own sex determination gene, then there is a lot of work to do. Yeah. So this is really surprising, and we don't understand why these genes are so variable. Okay, how much time do I have? Yeah. Yeah. Yeah, so I think I showed you that sort of genetic selection for changing these population sex ratios is only can only be done to a limited degree. And in this program that we are now funded by the FAO, we are looking whether we can use genetic modification to do this. And of course, I realize there's all sorts of issues with genetic modification, depending on where you're working, in Europe or not in Europe, or in another continent. But this is more like a proof of principle study to see whether we can use genetic modification to basically produce only males or only females of our insect of interest. So what we are doing is we are trying to knock out this male determining gene, so that males that carry it will not produce males anymore, because it becomes non-functional. And we are also trying to knock down or out this transformer gene, because if that one doesn't work, then we may not get any females anymore. So what we do is, this is the transformer gene. We just cut a piece out of it with the CRISPR technique, gene editing technique. I don't go into details. And our idea is, okay, let's make a non-functional transformer gene and a non-functional male determiner. So we have an individual that would be homozygous for this non-functional transformer, and no M, that would of course be a male, because you need the wild-type transformer to get this female loop starting, right? And then we thought, okay, if we can then make the females heterozygous and the males homozygous, then what will happen is this female, when she mates with a male like that, she produces half of the offspring that gets at least one wild-type transformer gene, which is active, and therefore you get females, and the other one, which pairs with another one for the male, which are both negative, or not wild-type or mutant, then you get a male. And this has been done already in the Mediterranean fruit fly, so you can do this. Basically. And then the idea is as follows. Okay, now if you have a strain like this, then you can just take any wild-type female, and you mate it with a male like that, and she produces always, because she's Tra plus, she always produces females, 100% females. Now, if you're not interested in 100% females, but for instance, you like to have 90% females, you just add 10% wild-type males, which have an M factor, and therefore these become males. Yeah, so this is sort of the principle, and that's what we are at the moment trying. While I speak, people in the lab are trying to make these knockout transformer genes. We have already managed to knock out the male determiner. So if this works, then we can basically produce any proportion of females that we're interested in the population. Okay, now I'd like to switch to another group of insects, and also to another application, which is biological control. And here, what we are interested in is having more females, because especially many biological control agents, not all of them, are parasitoid wasps. Parasitic wasps are traditionally used a lot. Sorry, there's an S missing there. In biological control, because females of parasitic wasps, they lay their eggs in other insects, either in the larval stage or the pupal stage of other insects, and then these the larvae of the wasps, they eat the insect, and of course then they kill the insect. So then instead of getting the original insect coming out, you get the wasps emerging out of that. Okay? So that's a very widespread technique. Big companies that produce lots and lots of these parasitic wasps for biological control, they're released into greenhouses and into other crops. And I think the biggest company is in fact in the Netherlands, near Rotterdam, which is a big family company that that has lots and lots, hundreds and hundreds of people employed and producing all sorts of different species for biological control. So how can we make more females in parasitic wasps? Well, here it's a bit easier. We are not so much constrained by the genetics, as I will show you in a moment. And we use this nice species, which is called Nasonia vitripennis, which is just a small parasitic wasp of 2 to 3 millimeters. So it's even smaller than a Drosophila fly. And this is our model organism that we use. So not a housefly, but a wasp. And this is the life cycle of this wasp. So this is a parasitoid of fly pupae. And these are different blowflies that, in fact, the larvae of these flies parasitize birds. And here you see, for instance, a small young bird with lots of these blood-sucking larvae on it. So this is a parasite of pupae of these flies. It is also used as a biological control agent, but there are better species to do this, in fact. And of course, here's then the life cycle of the wasp, which is again very short. So therefore, it's a very easy animal to work with in the laboratory, to breed it, to do genetics on it, and it has about a 14-day cycle. So here you see the larvae in the fly host. The larvae have eaten the fly that was in there, and then of course we get wasps coming out, and we get about 30 wasps out of one fly pupa. Now, I see the circles are shifted a little bit, but of course I just was talking about houseflies, and this is the insect group of the Diptera, right, which have these sex chromosomes. But as I said in the beginning, now we are looking at a hymenopteron, the ants, the bees, and the wasps, and they belong to this green group, and they have haplodiploid reproduction. And I have told you that then there are no sex chromosomes, and the only difference is females being diploid and males being haploid. So a female produces a haploid egg, and a male produces a haploid sperm by mitosis, because it cannot do meiosis, cannot reduce the number of chromosomes because it has only one. Of course, there are more chromosomes, but I just show you just one. And then what can happen if the egg is not fertilized? It remains haploid, and it becomes a male. If it is fertilized, it becomes diploid, and then it becomes a female. And I always say that males in hymenopterans are a bit a pity, because they only have a mother. I mean, they don't have a father. Yeah. So, and interestingly, when a female mates with a male, and this also, for instance, occurs in honeybees, she stores the sperm in what is called a spermatheca here, which is a very small vesicle near her oviduct. And basically, when she lays an egg here out of the ovaries, she can decide herself whether she adds a sperm to that egg or not. So she has control over the fertilization process. So she basically can decide herself whether she makes a daughter or a son. Okay? So in that way, she's much more flexible in changing the sex ratio of her offspring than a fly, because that always has to deal with the X and the Y chromosomes. Now, this idea was already, again, realized a long time ago, and this is what is called local mate competition theory, which was invented by Bill Hamilton, the late Bill Hamilton, in 1967 already. And he published this article on extraordinary sex ratios, and he say it has new implications for entomology. Well, he probably didn't realize how much implication this would have, because this technique of manipulating the number of males and females in the offspring is used a lot by biological control companies. And John Warren, which you see here, was sort of the first to test this theory using this wasp, Nasonia vitripennis. And what you see here is, if you vary the number of females that you put on say, on one host, then if she's alone, then she produces very few sons, because here's the proportion sons. But if there's more competing females, then you see that the sex ratio goes back to 50-50, which we call the Fisherian sex ratio. And these sex ratios we would sometimes call Hamilton sex ratios. So more females here than males if you only put very few wasps on a host. And I was really intrigued by this, because this is in 1983, and then I was a student. And I think I gave a talk on that, because I thought it was really, really exciting. And it was even more exciting that I then later on ended up as a PhD student at the University of Rochester. I was the first PhD student of Jack Warren. When he published this as a PhD student himself, he was still in Utah, but then he got a position in Rochester, and I became his first PhD student. And from that moment on, I've been, as you will see, been working with this wasp. Okay, so how can we change population sex ratios? Again, we have the same sort of gene regulation also in these wasps, so this is the same as in flies. But of course, there is no male determining gene here. And I'm going to go over this quickly. It turns out that the female silences this gene here in the zygote. So she imprints it, and you need a copy from the father that comes in via the sperm to start the female development. So it's now exactly switched, right? Because now you need a genome from the father to become a female, whereas in the flies, you needed it to become a male. We then looked for the gene that was responsible for this imprinting, and it must be realized that it should be a gene that is expressed earlier than the transformer gene. So this is hours after egg laying, and the gene sort of becomes active in the zygote around 6 hours after egg laying. It's also present in low levels already earlier, but that's the messenger RNAs that the mother had put into the egg. So this is the first transcription in the zygote of the transformer gene, and we knew that the transformer gene itself was not imprinted, but there must be another gene that is doing this. So we looked for a gene that was expressed even earlier than 6 hours after egg laying, and so this is one of the earliest genes that we know that is expressed in a zygote. And again, to make a long story short, we identified the gene, and we could show that it's only active in a fertilized egg and not in an unfertilized egg, because in an unfertilized egg there is no active paternal copy. We called the gene WOM. Originally we called it womanizer because it makes females. But because there was already a gene feminizer in honeybees, and we thought, okay, let's make a joke. Let's call it womanizer, because then it's feminizer in honeybees, which is a slightly different gene, and we call it womanizer. But then when we tried to publish this, we had a problem, because we tried to publish in an American journal, and they didn't like the word womanizer. So we had to change the name of the gene before we could publish. So then we came up with this name, wasp overruler of masculinization, which is basically the same thing, but because the abbreviation of the gene was already in the literature, so we did not want to give it yet another name, because that would be even more confusing. Again, very shortly, it turns out this is a new gene, not present outside Nasonia, the genus. It has some parts of genes that we know, P53. We also carry P53. But there's also a part in there which we call LOCK XX, and the name already says it. We don't know what it is. We cannot find it in any DNA database. It's a piece of DNA that is coding, and we don't know where it comes from, and we don't know what it is. If we look at the close relative again, it's not there. And so this is the regular, or it's a copy of P53, but it's not sex determining. So only by gaining this piece of DNA, it became sex determining, and only in the Nasonia genus. So again, the same story, it's a new gene, and it seems to be almost species specific. We looked a little bit at the origin of this gene, and again here you see that only womanizer is here present. So this is a phylogeny of the Hymenoptera. The honeybee is right here at the bottom. And there's this P53 gene, which is present in all of these Hymenopterans. Then that somehow became duplicated sometime. So there's some species that also have this duplication, which we call number 2. They also have number 1. But then number 2 became womanizer, or a wom, and these wasps also carry the original copy. So again, a new gene by duplication of an existing gene. Can I still, yeah? Last thing I like to talk about, and that is an observation that is already also quite well known, and that is that many insects are infected with endosymbionts that manipulate their reproduction. So endosymbionts are bacteria that live in the cells of these of the insects, and they cannot live sort of freely out in nature. They really are restricted to the environment of the insect cells or the arthropod cells, because there's many different groups that do this, so there's Wolbachia, which is the most well-known, but there's also Cardinium, Rickettsia, etc., and you find them in insects, but not just insects, also other arthropods, as you can see there. Okay, and they manipulate the reproduction of their host. Okay, and they are vertically transmitted. Okay, so they're only transmitted from mother to daughters, because they're transmitted through the egg. Because they live in the cytoplasm of the cells, and only eggs have cytoplasm. Sperm doesn't have any cytoplasm, so they're only transmitted from mothers to daughters. If they end up in a male, then it's a dead end basically, because they cannot be transmitted anymore. And you find them in all sorts of insects, and not just insects, as I said. It turned out that they can manipulate reproduction of arthropods in many different ways, and I cannot go into the details here. If you're interested in read some of this work from John Warren. And they can feminize, basically genotypic males, so they turn males into females. They can cause parthenogenesis. They can kill males in the offspring, and they can cause cytoplasmic incompatibility, and that's a bit more harder to explain, so I will not go into details. But what you should see here is that in all cases, they increase the proportion of daughters in the offspring. And from an evolutionary perspective, you can understand this because any mutant, any variant that is able to produce more, that induces its host to produce more daughters, will gain a transmission advantage over the ones that don't do it. Right? Because they're only transmitted to daughters. So they're basically parasitizing the host to produce more daughters, and therefore increasing their own transmission. So we are looking into how they do that, how they cause parthenogenesis. And now again back to one of the first slides. So we have sexual reproduction and asexual reproduction, or parthenogenesis, but in haplodiploids. And so this is a difficult term for sexual reproduction in a haplodiploid organism is aranotochy. Don't have to remember that. And if you're parthenogenetic and haplodiploid, you are, we call it thalitochy. Okay, so here we have males and females, but here we have only females. And it turns out that, okay, it's the Wolbachia, or the endosymbionts, not just Wolbachia, also other groups that cause this parthenogenesis. So females get infected with these endosymbionts, and then they simply become parthenogenetic. Well, simply, it's not so simple, because what we know is that these bacteria, they first cause diploidization. These females produce haploid eggs, and somehow the bacteria manipulate the egg in such a way that there's DNA replication, but there is no cell division. So they make the eggs diploid, and then it turns out that is not enough, because that would be a male. They also somehow have to feminize them, So you can only get daughters. And the bacteria are able to do that. We studied that in this species, Asubara japonica, which is from Japan, and we find both sexual and asexual populations. And this is in fact an interesting wasp because it's a parasitoid of Drosophila suzuki, which is a fruit pest all over the world now. And interestingly, what we can do is we can feed these wasps antibiotics in the laboratory, and then we kill the bacteria, and then they become sexual again, okay? So then we don't get the diploidization, the feminization. So we can cure them from their asexuality, basically. Okay? And so this is a beautiful system, because we basically have 3 different groups here, sexuals, asexuals, with the bacteria, and asexuals without the bacteria. And then my postdoc here, Elsa Meek, what she has been doing, she has been looking at how this transformer gene is expressed in the ovaries and the bodies of these wasps. So here you see these 3 different groups again. And basically what you have to get from this, it's a bit more complicated. There's also transformer 2 gene. Just look at this one. So here is the transformer, and here's the female specific splice form. And it turns out that if you're sexual in your ovaries, you don't put it in. The females don't put that in. But if you're asexual and you have the bacteria, this female transformer is put into the eggs. And if you remove the bacteria, it's not there. So it's the bacteria that are responsible for the female splicing of the host transformer gene. So they basically interfere with the sex determination of their host. And we're currently trying to find out how that may work, and we think there may be small RNAs involved, but unfortunately there are at least 550 small RNAs expressed in the ovaries, of which 64 map here to the genome of the Wolbachia. So yeah, we're not there yet to figure out how this works. Okay, yeah, this is then sort of my last slide. Of course, we like to use this information again to, if we can do that, can we transfer these bacteria to other species and make them parthenogenetic, because then they produce only females, and this may be, again, very useful for biological control. And what you see here is some articles that were published just recently, where people are already using these endosymbiotic bacteria for pest control, for mosquito control, but also for other pest control. So there is a lot of promise of using these endosymbionts for applications. And I think that, yeah, that brings me to my conclusions. So I think I've shown you that of course, most insects reproduce sexually with separate sexes, but that sex determination mechanisms are very diverse, and that it may be useful if we can sort of manipulate the population sex ratios for certain purposes. But there are limits, there are genetic limits. The sex chromosomes sort of genetically constrain sex ratio manipulation, and ook phenotypic and artificial selection have their limits. Haplodiploids are in that sense a bit more flexible, because their sex ratios depend on fertilization rather than on sex chromosomes. And with the last example, I hope I have shown you that also endosymbionts may be useful for application in pest control. And that brings me to my acknowledgements. Lots of people in the group that have, of course, done all this work over the years. Some international and other collaborators, as you can see here, and some of the funding that we received for this work. And that brings me to the questions, if you have any. Thank you. And I apologize for going over time a little bit. But
hello, thank you very much for your presentation. I just wanted to ask you for some study that report that they can found diploid males in parasitoid. How can this be possible?
Yes, that is caused by the sex determination system. This also happens in honeybees, but you don't find them in in nature because the workers recognize the diploid male larvae and kill them, but they are produced. And this is also occurs, for instance, in bumblebees and certain wasps and ants. They have a different sex determination mechanism than the Nasonia, which I've introduced here. And it's a mechanism that depends on homozygosity on a single locus. So if they have two identical alleles on a single locus, which is a sex-determining locus, then they become diploid males. So they have to have two separate alleles. So if there's a lot of inbreeding and there's a very few alleles left, then you get a lot of diploid males in these populations. Yes, that can also be a problem in biological control because some wasps also have this.
Yes. I was specifically talking about that because in breeding depression, that can be caused when you grew a lot of insects in laboratory conditions for a lot of generations.
Exactly. Yes. So inbreeding depression can be a problem with some of these also biological control cultures. So you have to sometimes sort of put in the fresh genetic material into these populations. Yes.
Okay. Thank you very much.
Leo, thank you very much for your talk. I have a question particularly regarding to your experience with the food industry. You explain us about the relation, how the sex determination and manipulation of the genes, so you can produce more females and more males. For food industry, for us, it's really interesting about the way in that we can make food. And this is a city that we can make a lot of flies because we have a lot. So, can you tell us about your experience with the way that you can produce food for people? So, how do you manage to connect with the industry? Probably the ideas could be help us to make more innovation project with that. We are not doing that. So, experience with the food industry. So my first collaborations on the food industry started with this Amuska company, but basically the director of that company contacted me because he saw that I was publishing on sex determination in houseflies, and so he contacted me, and then we started to talk. Okay, how can we do here something? Can we manipulate? And from that moment on, we managed to also got funding. So for a lot of this sort of a bit more applied funding, at least in our country, we need to have a certain percentage of private or governmental money. And so this company was willing to basically support applications. And in that way, that's sort of how I built it up. Do you have some recommendation for a student to how to go there, or for academics?
Yeah, I mean, many of these companies are startup companies, so I would almost say if you have a good idea, start your own company. But I mean, so this black soldier fly, say 99% of almost all of the larval production, feed production is done with the black soldier fly rather than the housefly. But there's hardly any genetics being done or selection yet. I mean, we're really at the start of that industry. But I think there's a lot of scope. I mean, we have been breeding cows and pigs for more than several centuries, right, and improving them. So I think there's a lot of scope for genetic improvement of insects for feed production. Also, for instance, for manipulating the nutritional composition of the larvae. You can make them more fat, or you can have them more carbohydrates in them. So yeah, if you have good ideas, I would, and of course you have to find some startup money. But yeah.
Thank you. More questions? Okay, there's one here. There's another one.
Hi, excellent talk. I really enjoy it. So I would like to know, so what is the factor of temperature? So do you need, for instance, for certain parthenogenesis at fixed temperature? Because you make the point that this occurs at 25 degrees, for instance, in the case of when it was determined that the first peak was for males and then for females, for instance.
Mm-hmm.
So what is the role of temperature in the fertilization itself, and the parthenogenesis that you show is depending of temperature? Do you need a special temperature to do that?
Not really, but the endosymbionts are temperature sensitive. So if you would give your wasp a heat shock, you may kill your endosymbionts. We use antibiotics, but you can also use heat shocks. But antibiotics are just a bit more efficient. So if you, in the case of endosymbiont-induced parthenogenesis, then temperature plays a role. But an egg can develop parthenogenetically both at low and high temperatures.
All right.
Yes, as far as we know. Yes. Mm-hmm.
Hello, thank you for your talk. Sorry. I have two questions.
Mm-hmm.
The first one is, well, first time I know about these high diversity ways of reproductions in insects, and I was wondering all the time why this happens. Because, I mean, in other organisms it doesn't happen, so what could be an explanation for this?
Yeah, I don't think we have a good answer to that. I mean, I'm an evolutionary biologist, so I also asked this why question. I tried to, but the how question is a bit easier to solve. Certain groups have this enormous diversity, not just insects. Fish are also very diverse in their sex-determining systems. For some reason, some groups are completely fixed. I mean, mammals are all XXXY, and yeah, there is no variation. Why a certain group still has so much variation and another group not? There's all sorts of theories about it, but there's very little empirical evidence for these theories, and it's also not so easy to test these theories, because you would have to sort of be able to manipulate the sex determination system to test whether indeed certain benefits or disadvantages or selective forces act on it. Mm-hmm. So I'm afraid it's something that I've been wondering about all my life, basically, since I started thinking about this, and there's still not a good answer to it.
Okay, thank you. And one short question, because I recently learned that these soldier fly,
black soldier flies, yes.
It's also here in Chile. This is worldwide distributed?
I think it's originally from the Central America, from the subtropical regions. So but it's now all over the world as well. I mean, it occurs now it's now an invasive species or an exotic species in the Netherlands, for instance. Of course, it did not occur in Europe, but now, of course, it escaped from these factories, and it establishes in nature. Mm-hmm. But I think it's originally from here, indeed.
Oh, okay.
So, yeah.
So invasive over there. Okay, yes. Thank you very much.
Yeah.
Thank you for your talk. It's an interesting theme, and the question is related with the temperature control. And I don't know nothing about that, but is possible that you control the sex differentiation when you change the temperature in the eggs, for example? It's similar with like, reptiles, for example.
Yeah. But I mean, reptiles and amphibians, they have really environmental sex determination, where really it's temperature, or sometimes it can also be another environmental factor that determines whether you become male or female. I mean, turtles, they lay their eggs in cold areas, they get more males, and in warm areas more females. But of course, in insects that does not really occur because, again, of this sex chromosome system. And so the segregation of the X and the Y is not temperature sensitive, it's just 50-50. It's genetically sort of determined. But it's an interesting question because we think that there is also a temperature effect in these houseflies on the strength at which the male and female determining genes are expressed. And we think, in fact, that may be one of the selective forces, to come back to the previous question, that may cause changes in sex determination. If temperature somehow affects the female determination function of a gene more or less than the male determining gene, then this may also sort of cause compensatory changes in the sex determination. But this is more speculation than that we have very good evidence for it. Yeah.
Okay, and just for curiosity, when you fed the chickens with these flies, you deliver it directly with the fly, kill fly, or you process the flies? For
No. I mean, we just feed the larvae live, because the chicken like to eat live food rather than meal or so. Yes, but of course they're also used to take proteins out, to purify proteins. And they're also thinking now more and more about technical products. So, like in the cosmetics, they can use certain compounds out of these larvae, and also in the glue industry and things like that. And in the paint industry, I think also. Yes. Okay. So they're looking for more and more applications of these larvae. Yeah. Mm-hmm.
Okay. There's one question at the end. Then another one.
Thanks for your talk. If we have a disequilibrium between the males and females, and you speak about a value, a social value, perhaps about in favor of one of the male or female, but I think that species have a multiple ecological service. So it could be good for a company to protect or to incentive a bigger number in female, and for others that have in observation another service could be the male.
Exactly.
So the final effect could be null, or who have the right? What arrive first to the system? I have problem with this. Perhaps one health vision about it. What is your opinion?
Actually, sure, but I got the question. Can you repeat the question, or maybe can you translate?
Okay, he's talking about the way that the company, one company could be protect or make more
okay.
health.
So it has to do with patents.
The way, yes. The way in the One Health view, what's your opinion on that?
Yeah, I mean, I'm not in favor of patenting new innovations in general, but I mean, I'm a scientist, so I don't need to care, I guess. But if you're a company and you find something out, then of course you may be interested in it. But I think breeding and insect strains cannot easily be patented, is what I understand from also my interactions with the company. That's apparently, that cannot be patented, as far as I know. But I don't know all over the world now. So the breeding, how you best breed them, that is sort of common knowledge. And nobody sort of patented any black soldier fly strain or something like that. I think, in fact, all over the world, there's only 2 strains that are being used, talking about problems with inbreeding and diversity. Yeah.
Christian.
I'm not sure whether that answers your question, but...
Okay. Thank you, Leo, for your very good presentation. I learned a lot about sex determination and the variation and the molecular mechanisms involved. My question is regarding the use of the manipulation of endosymbionts to produce asexual individuals, asexual offspring for releasing.
Mm-hmm.
But I wonder, perhaps I missed something during your presentation, but I wonder how can you manage the effect of parthenogenetic individuals? Because, as you know, many insect species are parthenogenetic. So if you produce parthenogenetic individuals for releasing them, probably the population will grow faster and may produce even more problems for agriculture, for example.
Well, I mean, I would not make the. Well, it depends on the purpose.
species.
Yeah, I mean, if you would make these houseflies parthenogenic, if that would be possible, then of course you don't want them to release from your factories. But I think the most prospects here are in parasitic wasps that then produce more females that are then released as biocontrol agents. So they are not pests, right? Because they are the ones that kill the pests.
Okay.
But that's for when they cause parthenogenesis. I showed you on one slide there are 4 different ways of how they can manipulate host reproduction, and another one is what is called cytoplasmic incompatibility, and that's what they use in the mosquito industry. So they release mosquitoes that are infected, and when they mate with mosquitoes in the field that are not infected, then they don't get any offspring because they cause an incompatibility. Okay, and yeah, in that way they do the first field releases on that. I think in Mexico and on certain islands as well already.
Okay.
Yeah.
Thank you.
Okay, I think we don't have more questions. Aquí, one more. Last one.
La pregunta, ante todo, muchas gracias por la presentación. Fue muy buena y aprendimos mucho de la reproducción de insectos y la determinación de genes. La pregunta es, en el contexto del calentamiento global, ¿las metodologías que usted explicó sobre el control biológico se pueden aplicar, ya que al haber un calentamiento en el ambiente y un aumento de la temperatura, el desarrollo de los insectos es mucho más rápido? ¿Se podrían utilizar para control biológico y se utilizan actualmente o son candidatas?
You have to
Okay, I will translate here. No problem with that. Well, he first thanks for the talk. Was very, very clear for him.
Thank
so
you.
he asked him about climate change. So if in now we are facing the increase in temperatures, so and the climate change are affecting us. So he said, if your methodology, all the methods that you're using, can be or could be stay in the same way that the climate change are changing, and you're using the same methodology, could be an effective way to treatment, or you have to make some changes. How is your experience with that?
So you're that's a difficult question. I mean, if I think about environmental sex determination, I mean, I think this is a concern with turtles and amphibians, right? That there's global temperature changes, and at some point the temperature at which one of the sex, at the lower temperature, is produced is not there anymore, right? And so people think they certain species that are temperature sensitive in their sex determination may actually go extinct because they cannot adapt. Yeah, I have not really thought about whether some of these techniques could help solving the effect of climate change on insects.
Or maybe the technique works for the purpose that you're doing.
Mm-hmm.
Now with more temperature or with climate change effect?
Yeah, but I mean, I think you must have a system that is temperature sensitive in sex determination to be affected. And yeah, maybe with these endosymbionts that could be an effect, because they are typically present in the warmer areas. I showed you this distribution of Asabaria japonica. So the sexuals are in the north of Japan, and the asexuals are in the south. So, and they are infected, and the ones in the north are not infected. So, in that sense, a change in temperature could change the distribution range of the infected individuals versus the sexuals. And at some point, the sexuals may just be disappear. Yeah. Mm-hmm. Yeah.
Okay.
I hope that gives some of an answer.
Okay. Okay, that's all. We would like to. Then I will talk in Spanish. Well, we would like to thank Leo for your amazing talk, and for us was fantastic having you here, at least for one day. Queremos agradecer también a todos los presentes por asistir a la charla de inauguración de nuestro programa de doctorado. Agradecer a los estudiantes que están algunos están acá presentes, pero no sé, lo vamos a conocer hoy, y otros están conectados en línea, ya que están llegando dentro de la próxima semana. Las clases parten la segunda semana de abril, y varios supieron que ganaron beca ayer. Entonces vienen en proceso y ya tienen comprados sus pasajes, ya nos avisaron que tienen comprados sus pasajes y están llegando a Chile la segunda semana de abril. Muchas gracias a todos por estar acá. Le damos un aplauso a Leo por su charla. Y vamos a hacer la entrega de un presente. Leo, we are now in a region that's particularly important for winery. So I'm not telling you that here's a wine here, probably you will see, but it's a gift for our university, for you. Thank you very much for being with us.
Thank you. I hope I can take it out in the
Thank you very much.
I'll drink it before. Thank you.
Muchas gracias a todos por asistir.